science relates?
Do certain tools and techniques that you use in lab remind you of common things you use and do every day?
For instance, when you use the vortex, do you think of using a blender at home?
What about the electrophoresis chamber? Does its bubbling remind you of a hot pot of water on your stovetop?
We will show you how often these scientific techniques imitate our everyday kitchen routine. A pipette is a laboratory instrument used to transport a measured volume of liquid. Pipettes come in several designs for various purposes with differing levels of accuracy and precision from single-piece glass pipettes to more complex adjustable or electronic pipettes.
Pipetting. As my partner and I demonstrate in the photosynthesis lab, a single-piece disposable pipette works by creating a vacuum-gathering liquid in the liquid-holding chamber. By selectively squeezing the bulb and releasing this vacuum, you may draw up and dispense liquid as needed.
For correct results, we only want the top dark green layer, not the lime green lower layer. Luckily, with this type of pipette, we can control the vacuum and extract only the layers that we need for the lab.
But how does pipetting actually work in the kitchen?
How this yummy procedure works. In the kitchen, we used a baster like we used a pipette in lab. Basters are used to extract juices or marinades onto meats. We used a chicken. But hopefully, you all brought y'all's basters for this coming turkey season.
Micropipetting.
Before you can start pipetting, you first grab a sterile disposable tip by applying pressure downwards onto the tray into the tip. And now, you're ready!
Insert the tip into the container you wish to pipette from. As demonstrated on the pipette, you push through the stop and then release the hold to extract the liquid. The liquid remains in the tip until the next time in which you push through the stop. By doing so, you eject the pipette's contents.
Here as shown, this is an example of not pushing through the stop. As you can see, there is liquid still in the tip. For your experiment, this creates inaccurate results. So you want to make sure to push through the stop to dispense all the liquid from the tip.
Spectrophotometer. Remember, once you turn on the spectrophotometer, you must wait 15 minutes for the machine to warm up. With the wavelength already set, you may put the sample test tube called a blank into the spectrophotometer. Close the hatch, then set the machine to 0. Afterwards, once that is done, you may remove the blank and insert the tube in which you want to be read into the spectrophotometer. And now you're set. In case of any confusion, the instructions are set on the spectrophotometer.
Dye-less tubing is a semi-permeable membrane used in our labs to facilitate diffusion or osmosis. First, we must prepare the dye-less tubing by retrieving the desired amount of solution. In our case, it's 10% NaCl. Then, you must add your solution to the dye-less tubing with one end clamped off. As we did, you may find this to be difficult. So feel free to use your lab partner's assistance. Once the tube is open, pour all of the solution into the dye-less tubing. You must then proceed to clamp off the open end. Fold the tubing until the tubing is taut. Finally, place the folded end into the clamp, and then you're ready for diffusion. Now you can't tell me that I have dye-less tubing in my kitchen.
Yes, I can. Cheesecloth.
Cheesecloth?
Yeah, cheesecloth. It's a selectively permeable membrane found in the kitchen. Use it in cheese making, straining stocks, bundling herbs, making tofu, thickening yogurt.
Oh, well now I see how cheesecloth and dialysis tubing relate.
Using the spinning probe for mixing to ensure diffusion by dialysis tubing.
As you see, the dialysis tubing is inserted incorrectly into the beaker. You must put the dialysis tubing in the correct way as shown, in order to allow the probe freedom to spin. Then set the RPMs for the probe to spin. Wait, now I can see the dialysis tubing and the cheesecloth relating, but how does the probe work in the kitchen?
Well, you see, the spinning probe is much like a whisk. It mixes around the contents in the beaker, like here shown in the picture. Mortar and pestle. Mortars and pestles have been used for several years, not only dating back in old science labs as well as now today. They're used in kitchens and in labs. They're used to make medicines also.
During the photosynthesis lab, we used the mortar and pestle. First we cut up the leaves and then we added sand to recreate the friction.
Using this sand, we got to grind up the spinach leaves until the desired consistency. We want it to be a liquid, that way we can pipette it later. Mortar and pestles are used to grind up spices. We commonly see it in kitchens for that purpose, but it's also used to make certain juices, like olive oils. We see it here commonly in the mocajeque to make salsa.
In the kitchen, we used a mortar and pestle to grind up garlic as shown. That way we can make a yummy garlic butter.
As I said earlier, after we ground it, we got to the desired consistency we needed to use our pipetting techniques to use it later in lab.
In the same lab, we used a vortex to mix up the liquid. We couldn't help but notice how the vortex reminded us of a blender.
This whirling motion, in which we could see better in the blender, is called a vortex. That's why it gets its name. Using the analytical balance. Luckily, the analytical balance has an overhead door and a side door. We preferred using the side door. You must first turn on the machine. Afterwards, open the side door. Insert your parchment paper or perhaps your tray in which you'll be loading your contents. Then you'll notice that it is measuring. You want to zero it out by pressing the O/T button. After zeroing it out, you are able just to measure the dry ingredients. Jamie then added more dry ingredients. You have to be very precise and careful not to add too much. Any 0.1 of a gram over would be too much and therefore lead to inaccurate results.
That's right, Lanny, it's kitchen time!
For sure. Many people don't have an analytical balance at home, but I know for a fact I have a scale at my home. We commonly use it in many cases when we were using cooking recipes. Like, for example, when we make pies this Thanksgiving, yeah.
Here we measured a powder with a kitchen scale. First we zeroed the scale out so it wouldn't measure the bowl, just like we did with the analytical balance.
Casting a gel. Casting a gel for DNA fingerprinting. After checking that the temperature of the gel was around 65 degrees, to make sure that it doesn't set too early or too slow, we then poured the gel into each individual tray, making sure carefully that it doesn't overpour. Then we proceeded to the second tray and did the same. We did find pouring the gel to be a little bit time-consuming because we had to be so meticulous and precise to make sure it didn't overpour. But we found it even more time-consuming waiting for the gelatin to set. We couldn't help but fidgeting.
After our lab, we decided to make a jello of our own. So we also had to pour very carefully to cast this gel. Can you guess what this green gel is?
Careful, Jamie. Watch out. Make sure you don't overpour. Still can't figure out what this green gelatin is? It's Jell-O! That's right. Our green gelatin was Jell-O, and I know you've got to have that in your kitchen. After casting your gelatin for DNA fingerprinting, you must make sure before the gelatin actually sets to insert your comb. These will be your analysis slots in which you will micropipette the DNA later.
Once the combs are in place, let the gel set. Then you must remove the combs before electrically charging the gels. We thought the bubbling was really cool. It reminded us of a pot of hot boiling water. Now do you see how science and food relates?
We've shown you the most common techniques in our biology lab and how they mimic your kitchen habits while you're cooking something up.
Perhaps now that you know how often you use these science in your everyday life, you will start to see similarities between our biology lab sessions and what you use at home.